LSD, lysergic acid diethylamide, is a potent hallucinogenic drug that has a profound impact on perception, mood, and cognition. Commonly referred to as acid, LSD is classified as a psychedelic and is known for its ability to induce altered states of consciousness, vivid hallucinations, and sensory distortions. It was first synthesized by a Swiss chemist named Albert Hofmann in 1938, but its psychoactive properties were yet to be discovered until 1943. LSD is derived from ergotamine, an alkaloid produced by the ergo fungus, which commonly infects rye and other grains. This fungus has historically been known for its toxic properties, but it was the discovery of ergotamine and its subsequent transformation into LSD that led to its use as a psychedelic compound. The chemical structure of LSD is unique and complex. Its molecular formula is C20, H25, and 3O, which consists of a lysergic acid core structure with a dithylamide group attached at the nitrogen atom. This structure contains an indolate ring, a feature it shares with serotonin. Serotonin is a neurotransmitter that plays a key role in mood regulation and perception. This structural similarity to serotonin is thought to explain the drug's ability to interact with serotonin receptors in the brain, leading to its profound psychoactive effects. The amide group attached to the lysergic acid backbone plays a significant role in the molecule's lipophilicity, which enables LSD to easily cross the blood-brain barrier and exert its effects on the CNS, the central nervous system made up of the brain and spinal cord. Additionally, the rigidity of the fused ring system and LSD structure contributes to its biological effects and activity and potency as a hallucinogen. LSD is synthesized through a series of chemical reactions that begin with the alkaloid ergotamine, which, as previously mentioned, is obtained from the ergo fungus. The first step in the synthesis involves the hydrolysis of ergotamine using a strong acid, such as sulfuric acid or hydrochloric acid. The ergotamine is hydrolyzed in order to produce lysergic acid, and during this process, the ester bond between the ethyl group of ergotamine and the rest of the molecule is broken, releasing lysergic acid as a free compound. Lysergic acid is the core structure that will undergo further modification in order to produce LSD. Once lysergic acid is isolated, the second step in the synthesis involves a condensation reaction with dithylamine, a simple amine. In this step, the dithylamine acts as a nucleophile, attacking the carboxyl group of lysergic acid to form an amide bond. The reaction forms lysergic acid dithylamide, LSD, which is the desired psychedelic compound. This reaction is typically catalyzed by acid chlorides or carbamides, which facilitate the formation of the amide bond by activating the carboxyl group. Once the LSD is formed, the final step in the synthesis is purification. The product is purified through a process called recrystallization, where the LSD is dissolved in an appropriate organic solvent, and the solvent is gradually evaporated to isolate the pure crystalline form of LSD. This results in typically a white, odorless, crystalline powder. The purity of LSD is crucial, as even small impurities can affect its potency and the nature of its effects on the user. LSD synthesis is simple, but it requires a careful control of conditions to ensure high purity and potency. Understanding the chemistry behind LSD synthesis is able to provide insight into its dramatic effects on the brain. Now that we know what it is and how it's made, we can talk about the biological effects of LSD and how exactly it interacts with the human body. Once consumed, LSD quickly finds the brain's neurotransmitters, specifically the serotonin receptors. It binds to these receptors and mimics the effects of serotonin, but in a more dramatic and disruptive way. The cerebral cortex becomes highly stimulated, which leads to hallucinations and psychedelic experiences. These experiences include visual distortions where colors are enhanced, the illusion of geometric patterns, and inanimate objects moving and becoming lifelike. These effects occur because LSD disrupts the brain's ability to process and integrate sensory information correctly, which consequently blurs the lines between perception and reality. LSD also carries emotional effects, such as highs, like euphoria, empathy, and creativity, and lows, like anxiety, paranoia, panic attacks, and in extreme cases, psychosis. The effects of LSD can last up to 12 hours, and the aftermath usually leaves individuals feeling tired and disconnected from reality. LSD's biological effects are extremely complex and may never be fully understood. Personally, it is not a drug that I would ever use or recommend to anybody, but it was definitely very interesting to research. Thank you.